Thank you all for the invitation. Thank you for those who are in the audience. And of course, I truly enjoyed all of the talks that came before us, my presentation. So Neuraxem is a predictive and precision functional genomic brain medicine company. By functional, I mean we operate at a very different level. We operate at the level of gene regulatory networks. Our goals are early diagnostics and treatment of human brain diseases and disorders. We also have the privilege of being a spoke company in ARPA-H, which is a high-risk federal agency. The problem, as many have identified, that there were no early diagnostics or effective disease-modifying therapeutics for brain disorders and diseases. And one of the deadly assumptions for a long, long time was rodents were equivalent to humans. They're not, and many now recognize that. But we recognized that way back in the early 2017-18 timeframe. So, sorry, 2007. And in 2015, we disclosed for the first time that we had made a human brain model. It now has a US patent. The brain model is complete. And in 2023, the FDA has the Modernization Act, no animal testing. They want diversity in clinical trials. We're very well set up to do that. So our proprietary process makes these brain organoids, and they have everything from the spinal cord to the retina, all in one single entity. It has all the main brain regions. It has all the cell types, and is approximately 98% identical to the adult human brain. Just to show you some immunohistology very quickly, we can see all the parts of the brain with immunohistology. We even have the blood-brain barrier and the choroid plexus. And this becomes very important, as you will see shortly, and the microglia, this becomes very important to truly understand what is driving Alzheimer's disease. So just to give you an idea, we try to put precise in our precision medicine claims. Here is data. I'm showing you data. That is the development of the organoids. And they go from day 0 to week 12. You can see the identity plots. The R-squared coefficient here is 0.94 and above. And what you're seeing on each axis are replicates from about 100 to 200 organoids in a dish. Very, very replicable, very robust. We also do a high-field NMR for metabolomics. You can see the two traces. This is from an Alzheimer's brain organoid. The red and the blue ones are replicates from a 100 organoid batch. And on the right are just the plots, the quantitative plots. So once we have that, we can now go from skin cells to organoids, from organoids to big data, from big data, both metabolics as well as transcriptomics, create data soft banks, do the GNRL data analysis, and we can get early diagnostic markers, drug targets, and even drugs. Just to give you an idea, our portfolio is very extensive. We have made all of the organoids, and we have been funded for the opioid crisis by the HEAL initiative from NIH and for PTSD from the Department of Defense. Just to give you an example, I'm just going to quickly walk you through the data. You can come back to it later. We took Alzheimer's disease. Our data is heavily corroborated by postmortem analysis with a p-value of minus 100. It's independently corroborated by 80 genetics, which is GWAS studies. And we have now decided on the actual upstream targets of inflammation and lymphatic system dysfunction. And shown below here are the biomarkers that are dysregulated for inflammation and lymphatics. And there are two therapeutic targets. We have used a drug combination. One is an agonist against the first target, and the other one is an inhibitor against the second target. And when we do that, we completely reverse the inflammatory biomarkers shown here. I'm just using red and green to give you a quick preview. You can look at the data later on. It's the p-value of our analysis. We show that we also reverse the lymphatics that I pointed out to. And finally, we also reverse virtually all of the known biomarkers that are linked to Alzheimer's disease: BACE, APP, ApoE. MME is nephrolysin, which is involved in the degradation of the cleavage of A beta. So what are we now looking at? We're looking at novel therapeutics for AD. We have similar things going on for PD and aging, novel diagnostics. We have issued and provisional patents. We're getting very big into AI, ML, licensable brain disease portfolios for all the disease we showed. And as I mentioned, we are previously funded by NIH and DoD. Our market size is huge, particularly if you start to include AI and ML applications for in silico ADME talks and other applications and the drugs. I would like to make a very exciting announcement today that we are now going to be backed by a private trust for $900 million at 90%. We're going for unicorn status. The opportunity to join us for raising an additional $100 million is still open. And then finally, just to tell you our business model, we do scaling and validation, early diagnostics, and therapeutics. Our revenue streams come from the licensing of the platform, of the targets, and drug discovery, as well as the drugs themselves. And for all of the platform diseases, portfolio diseases I showed you. And finally, we have a lot of patents that have either been issued, filed, or in preparation. And these are our incredible people involved. I want to point out Dr. Peter Cruz. He's a member of the Royal Society of Medicine. And he is also our incoming CMO. And then there's Joseph Armstrong, his ex-Merck, will help us with the drug development. And all of the other ones have been pioneers in the beginning, which was nearly 10 years ago. And with that, I'll just stop and take questions.
All right. Who's got the first question?
I have a question. What are the what are the current limitation of AI and ML and data needed to guide its use in brain medicine?
Yeah, so the current problems of using AI ML, I come from the world of chemical physics and quantum physics. I understand this very well. It's really about having very robust, reliable, and rigorous data as inputs to teach AI and ML literally to become intelligent. In the absence of which, AI will never be intelligent. And that's why I showed you some graphical data to show you that we have precision, which is the replicability and reliability. I also showed you accuracy, which is the ability to recapitulate the human disease for Alzheimer's. We have very similar data for Parkinson's and many of the other diseases that I didn't have time to show you. So all in all, we are very well set up to work with in silico algorithms to create an entirely new virtual platform that'll be very saleable and marketable to big pharma or with lots and lots of other applications.
Thank you.
I have a quick question, Rene. Thanks for the presentation.
Sure.
Could you elaborate a little bit on your market exactly? Because it's obviously a big market with Parkinson's, Alzheimer's. But what customers are you addressing in your market analysis there? It's not the therapeutic.
Yes. So they depend on what part of our portfolio. For example, the drugs and targets and the use of our patented model to create drugs and identify therapeutic targets, validate drugs, and also validate their therapeutic efficacy is one market. But the other big market we are now looking at is the large companies like Google, Microsoft, Oracle, and NVIDIA, all of whom are getting very heavily into healthcare and the ability to do a lot of this stuff in silico. So what we're able to provide them with is a reliable data input and the output that'll drive their ability to do a lot of this stuff for the future, stratification of patients, looking at a lot of different things with our data input to do things virtually with AI ML applications.
So it's more representative of preclinical development dollars.
Yes, we are now in a capacity to do a lot of this stuff ourselves because, as you might have seen, we now have a backer that is backing us close to $1 billion. So we have our own internal teams, but we're also looking at external collaborators to augment what we need and push this. So the investment opportunity is huge for those who want to join in for raising the rest of the $100 million to get our unicorn status. So I'm excited. This work is, as you can imagine, is over 15 years old. We made the first brain organoid in 2015. We were heavily covered with it. And now we're just showing you how we have taken it into application. And I gave you one example with Alzheimer's.
Thank you.
One more question.
Yeah, thank you for the presentation. Maybe following up on this market segmentation, there's a whole bunch of lines of business here. And as you start to develop the business, is there one in particular? Is it the organoids as a platform for drug discovery for other companies? Is it the AI? If you had to rank stack all the lines of business, what would you really hang your hat on across this whole portfolio?
Yes. So if you think about where we want to go, actually, believe it or not, aging is going to be our biggest market, the first priority. The second one will be AI, ML applications for the IT sector. And then third, fourth, and fifth will come, the degenerative diseases, and then all of the others will follow. So there's a lot of opportunity to license things out. For rare diseases, we will license the platform, develop the models. We had a nice talk by Gilda Evans on autism. We have been involved in autism for a very long time. You can go back and look at me. I've been involved with Autism Speaks at the very beginning. So I just think that what you're seeing is a massive, massive opportunity that spans so many pivots or markets that I would be, I'm setting priorities based on what I think is the first to market and first to bring something into application, and then prioritize all the rest. A typical clinical trial will take you a little longer. It'll take you 5 to 6 years to get through phase 1 through phase 3. So we want to have marketable products. Diagnostics, for example, we'll bring it to market within 2 years. Early diagnostics, we already have it all worked out for Alzheimer's. We can stratify patients. We can do all kinds of cool things. And we can detect this decades and decades before anybody else can do it using any other technology that has blood-based technologies and others that people have brought to that you have seen being very, very heavily advertised.
All right, Norstim, thank you very much.
Thank you much.